Hoje a palestra da disciplina Seminários de Zoologia do curso de pós-graduação em Zoologia do Museu Nacional e do curso de graduação em Ciências Biológicas, ambos da UFRJ, será sobre a origem dos animais e ministrada por um dos maiores especialistas do tema, o professor Ken Halanych, da Universidade de Auburn, no Alabama, nos Estados Unidos. So it's a great honor to have Dr. Ken Halanych here with us to talk about metazoa evolution. Thank you very much for accepting the invitation. Dr. Halanych is a full professor at Auburn University in the United States. Along his career, he has received teaching and research awards and guided several undergraduate, graduate, and postdoctoral researchers and visiting scholars. Halanych has an outstanding career with close to 200 peer-reviewed publications, most in high-impact journals such as Science, Evolution and Development, Annual Reviews of Ecology and Systematics, and Systematic Biology. He is the editor-in-chief of the Biological Bulletin and a leading expert on origin and evolution of metazoa and marine invertebrates, Antarctic phylogeography, molecular systematics and phylogenomics, and chemosynthetic fauna. Without further ado, we have here, please welcome Dr. Ken Halanych.
So thank you, Joanna. That was a very nice introduction. I am very happy to join you all. It'll be fun to talk a little bit about what's happened near the base of the animal tree. This is somewhat informal, and I know we have both undergraduates and graduates. So if I start talking too fast and I need to slow down, please just put something in the chat, and then Joanna, let me know to slow down if that works. So this has been a hot issue in systematics, and it really came to a head or became a very big topic around 2014, 2015, 2016. But the origins of this debate go back a little bit farther. The first paper that really sort of suggested it with molecular data in any significant way was by Dunn et al. In 2008, but they sort of downplayed it. And then there was a subsequent paper in 2009 by basically the same author group with Andreas Henuel as the lead, which they dove into this issue a little bit more and looked at it. When we think about this issue, what we're really, sorry, now I am having technical issues because my slides don't advance. All right, there we go. When we're talking about the base of the animal tree, we're really dealing with 5 extant lineages. Of course, there have been a bunch of other lineages which are now long extinct, but this is what we have to work with today. So there are a couple groups, and some of these you know very well. There are sponges, which traditionally have been viewed as the first branch off the animal tree. We have ctenophores, which is some of the more recent molecular data propose are sister group to all other animals. And then some of the other groups of interest are cnidarians, so things like corals and jellies. We also have placozoa. This is a group that is a smaller, lesser known group. There's pretty much one nominal species, but we know there is quite a bit of genetic diversity. And there are about, I guess, 20 or 30 different lineages, but they all have diversified very recently. So it is a challenge systematically because it is a very long branch. And then to sort of round it out, bilaterians. We know bilaterians aren't near the base of the tree, but when you're trying to figure this issue out, you sort of need to include them in. I will tell you there have been big arguments, especially for Porifera, Cnidaria, or Placozoa as being the sister group to all other animals. And the recent debate's really between sponges and ctenophores. Some of some of what I've gotten interested in, and sort of one of the reasons this is a big question, is, so that we can sort of reconstruct and try to understand what the earliest animals might have been like. But what sort of got me into it was thinking about neural evolution. So some of my work on this was actually funded by NASA, the National Atmospheric and Science Administration in the USA. And what they were interested in was looking at how easy it is for advanced life to evolve. And what they mean by advanced life is multicellular life. And they usually mean life that can react to things. And so the way, as we know, the way animals react is by having nerves, and we can react pretty quickly to stimuli. And so there are some basic questions like, given the presence of life, what is the chance a nervous-like system will evolve? Can neurons or nerves evolve multiple times? Now, those are slightly different questions. So we know nervous systems and the organization of nervous systems have probably evolved multiple times. And there's not a lot of debate about that. People can understand that neurons sort of over evolutionarily time becoming progressively organized into nerve cords has probably happened more than once. The real debate is whether neurons, the nerve cells, whether they have evolved more than once. And then the other question here is, what factors favor the evolution of neurons and nervous systems? Now, at the heart of the sponge ctenophore debate, it really impacts how you answer a couple of these different questions about whether nerves evolved once or multiple times, and what factors might shape the evolution of nerves. So I'm not going to go through the full history of all this, because I want to have time to have this be a little bit more of a discussion. But so I'll jump right in. I already mentioned that there were papers in 2008, 2009, which suggested ctenophores were sister to all other animals. That was followed on around late 2009 by a paper by Hervé Philippe, which also used transcriptomes and genomic methods, and it suggested that sponges were the sister group. So it was a back and forth. And it really was sort of quiet until 2013, 2014. So the Ryan et al. Paper came out in Science in, I think, like November, December of 13. The Morose et al. Paper came out in Nature in, I think, January or February of 14. So these two papers were pretty close in time. And what they both did is they looked at the genome of ctenophores. The genomes of ctenophores are actually, at least these two species, are fairly small. And these are probably the best studied, the most studied ctenophores around. Not a lot of work has been done on ctenophores, but of the ctenophores work that's been done on, these two are pretty well understood. Now, to sort of cut to the chase, both of these papers, two independent groups, sequenced the genomes. They pulled together genome and transcriptome information, and they both basically came to the same conclusion. So both of them placed ctenophores at the base, or not at the base, as sister group to all other animals. So ctenophores, then sponges, then placozoa, then cnidarians. It's sort of turned around here, but it's the same basic relationship. Ctenophores, then sponges, then placozoa, and then cnidarians. There are a couple things about these trees. They're based on transcriptomes and genomes, so they have a lot of loci. Over about the past 10 to 12 years, this field has really evolved in a number of ways. So one of the things I like to tell people is that I started my career as a systematist back in the late 80s, early 90s. And when we were looking at single genes, there were all these papers about all the different types of mistakes you can make with molecular phylogeny. Maybe you don't have enough taxa, maybe you haven't analyzed it right, maybe you've used the wrong model of evolution. All of these arguments happened all over again once people started looking at transcriptome or genome data where they were looking at hundreds of genes. And so a lot of the argument about whether sponges or ctenophores are basal really gets into nitty-gritty details about methods, and I'll explain more of that in a second. But the important thing here is to realize some of the implications of this result. So, as I'm sure you all know, we do not think of sponges as having nerves or muscles. We also don't think of Placozoa, this one little oddball group. They only have, I think it's about 8 cell types. They are very organizationally simplistic, and they do not have nerves or muscles. In contrast, of course, we think cnidarians, and bilaterians have nerves and muscles. It's usually a lot better organized in nerves and in bilaterians. The nerves are really organized into good, for many bilaterians, are organized into good nerve cords and a very clear nervous system. In cnidaria, we talk about a nerve net that is more diffuse. And sort of the same with muscles. There's no question that cnidarians have muscles, they can contract. I'm sure you've all seen images of like a sea anemone that can squish down, and that is all muscle-based. Ctenophores are a little bit different. They also have nerves and muscles. Again, it is not well organized. But the debate with whether ctenophores are a sister lineage or sponges are a sister lineage to the rest of animals really has caused a lot of discussion because of what people suggest it means about whether nerves and muscles have evolved twice in animals or whether or not nerves and muscles evolved once. And if you move, obviously, if you move ctenophores over it, if you move ctenophores over into this region of the tree, you could very easily have just one evolution of nerves and muscles, right? So that's that. As I've already mentioned, I'll get more into some of the evolution of nerves, and not so much muscles, but nerves in a minute. But as I mentioned, really starting a little bit with the 2008, 2009 papers, but especially starting in about 2014, there has been this back and forth publishing of papers about whether sponges are sister to the rest of animals or whether ctenophores are. The paper you read for class sort of highlights some of that. So the editor of Nature reached out to one group who was publishing on the idea that sisters were, that sponges were sister to everybody else, and one group that was writing on ctenophore sister to everybody else, and basically tried to put us head to head so we could get our ideas out there and really help the field move forward. It's been very hotly debated. In some cases, a little too hotly debated. Without naming names, I will say that after some of the papers came out, people would come out and in the common press make statements like, oh, this problem is clearly solved. My work put an end to it. We know the answer now. And that just wasn't right. They took very strong stands and were convinced in their own work, which is usually not a good sign when scientists do that. Since you all are early in your careers, I will tell you that. But a lot of what it has come down to is issues with methods, maybe a little bit of issues with taxon sampling. And so most of the work since then has really focused on very, very detailed or very specific issues with things like how you conduct phylogenetic analyses. To sort of get at some of that, I had a postdoc in my lab, Nathan Whalen, who went in and we dissected the data sets a number of different ways. So we generated a full data set very similar to what the genome papers had done. And then we walked through and applied different types of filters to control different aspects about the data set. So for example, we did things like remove taxa that might be problematic with long branch lengths. We also removed taxa that were very slowly evolving, or we removed taxa that were very quickly evolving. Since Torsten is on the line, I will also give a shout out to him because a lot of the way we measured this was using some of the programs that Torsten developed to look at how taxa behave in a genetic data set. When we controlled for all of these things, we still found out that most of our analyses resulted in ctenophores being sister group to everybody else. But there were certain cases where that was not true. There were certain cases where sponges came out sister to everybody else. Now, after this paper came out, again, it was back and forth. So there was another paper by Pisani et al., and they're one of the groups that came out and sort of said, well, no, it's definitely ctenophores. But their analyses, I would personally argue, was a little more limited. They sort of selectively picked and reanalyzed data sets from our paper and from a earlier paper by Joe Ryan's group. But one of the things that came up and was a big point of argument or a big point of difference is that whether people use Bayesian analysis or whether people use likelihood seem to be favoring different trees. So a lot of the analyses that were run with Bayesian seem to be favoring sponges as a sister to everybody else. Those that were doing maximum likelihood seemed to favor ctenophores. Now, it turns out to get a little more complicated because the main Bayesian program being used at the time, PhyloBayes, employed very sophisticated models of amino acid evolution. So all of these trees that are being done, everybody's using amino acids because they're more conservative than the nucleotides. And the events we're talking about happened well into the Precambrian. They probably happened well over 600 million years ago. The challenge was, and what some of what Nathan Whalen, my postdoc, discovered is that the methods that were being used by this PhyloBayes program were doing what was called over-parameterizing the data. So with molecular data, you're looking at different parameters, you're looking at rates of change, you're looking at things like gamma distribution to understand how independent your sites are. You're looking at the frequencies of different amino acids. So you can add all these different parameters into your evolutionary model. The more parameters you add, the more complicated the model becomes, the harder it is to calculate the values for the model. And that's what seemed to be happening in PhyloBayes. And then the other thing that was a big no-no was when you run a Bayesian analysis, you have to run it because you're doing multiple chains or you're doing simultaneous runs. You need to run it to where those chains do what is called converging. In other words, the likelihood scores of those all those different analyses you're running within a Bayesian analysis converge to a similar point. It turned out many of the trees that were well, not many. Some authors chose that when they did phylobase runs, they would not run the Bayesian analysis to that consensus point. And so the analyses were being completed prematurely, which probably also turns out to be an issue in this. Okay, so again, highly technical stuff. I can answer more questions about that later. There's a back and forth. One of the more recent analyses was a 2007 analysis. I guess I shouldn't say that's the most recent. One of the more complete. There have actually now been sort of a bunch of analyses coming out. I may be a little biased, but I think more of them have been supporting ctenophore sister than sponge's sister. Okay, so let's talk about some of the ideas behind sponges being sister to everybody else. The idea that sponges were the first group of animals off the animal tree has been taught for hundreds of years, very well ingrained. It's one of these things that everybody just absolutely knew was correct. For those of you less familiar with English, a curmudgeon is somebody who is always cynical and critical and sort of has a glass half empty view of life. So they're always going to question things very critically. So people have argued choanocytes show that sponges are sister to other animals. Well, if we think about that statement, you know, you even go into invertebrate textbooks and they make statements like that, that choanoflagellates is a likely ancestor group from which metazoans arose. They possess these cells identical to sponges. There are several problems with statements like this. One, if you take it at face value, if sponges arose from choanoflagellates, you would expect that sponges were not just sister group to choanoflagellates, but were nested within choanoflagellates. We don't see that. We know that the choanoflagellates, we know that choanoflagellates forms its own group that's monophyletic, and we don't have animals coming out of the center of choanoflagellates. If you're going to count a choanocyte, the cell type, as a synapomorphy of the group, this is what we would expect the tree to look like. And we don't see that. What we do see is either this situation where ctenophores are sister to everybody else, or this situation where sponges are sister to everybody else. If we go just on simple parsimony, yes, sponges sister is more parsimonious, but it's only because of an additional loss, right? It's one additional loss. The other thing is that the comment that choanoflagellate-like cells or choanocytes have evolved only once is simply not correct. If you look across animals, you will find at multiple places in animal evolution, you will find a cell that has a long flagellum and surrounded by a collar, very similar to choanocytes. People have even talked about this in the case of like the cells that we have in our inner ear, which have a somewhat similar morphology. There's also more recently out of Sally Lee's lab, who is a sponge expert, there was work done studying choanoflagellates of sponges and choanocytes. And Sally's lab actually pointed out that there were several morphological differences between these two. So they're not as similar as everybody has seemed to argue. Okay, another point is I've heard people argue the ctenophore sister hypothesis is not consistent with the fossil record. We have very clear ctenophores going back to the Cambrian. So this is a little bit different than modern ones, but you can see it, what have been interpreted as teen rows, or these paddle-like cilia, which are unique to ctenophores. But there's also older fossils going back into the late Ediacaran. There are fossils going back to the late Ediacaran, which have been interpreted as ctenophores. Sponges are a little bit more interesting. There are clearly sponges going back to about 550 million years ago. There have been several papers that argued that sponges go back much farther, that go back to over 600 million years ago, but it is not totally clear what's going on there. Some of those sponge papers have looked at spicules. Fossil evidence right now is still very interesting in that if you follow the literature to some degree, you'll see that every so often there is a new fossil coming out that's pushing the date of a particular group back another 10 million years, another 20 million years. So I might argue with the fossil record that we're still waiting to find out what the fossil record says, because there is still a lot of new information coming out about this. Okay, I've also heard people argue that sponges lack ionic control. And there's the assumption that the basal lamina could not have been lost. I had this argument with a famous biologist in my living room one day. Anyway, it turns out that, again, a sponge lab has argued quite the opposite. Sally Lees and Ragoso have made the arguments that all sponges actually have a sealing epithelia. It may be a little different than a basal lamina, but they argue that sponges have ionic control. One of the other things that people may not realize is sponges can actually contract. The way they contract is a little bit different than a standard muscle cell, but they clearly have the ability to contract. And this has been best shown in freshwater sponges. And part of the reason people think they do this is that when their channels get clogged, if they can contract, they can basically clear their channels. Okay, how am I doing on time, Joanna? Okay. I've also heard arguments that ctenophores are too complex to be basal, and that basically sponges have to be the sister group because sponges are a lot more simply organized, and no doubt the first animals had to be simply organized. It just has to be that way. Well, complexity is driven by selection on function and is not constrained by phylogenetic history. We see this over and over again, especially when you start talking about parasites. When you get things that become parasites, or especially become internal parasites, they can lose all their complexity very easily. The other flaw in this argument is that modern day sponges and modern day ctenophores are over 600 million years removed from their ancestors. So it is not at all clear, at least to me, that a modern ctenophore or a modern sponge looks anything like or acts like what a sponge or a ctenophore did 600 million years ago, or what their ancestors, I should say, acted like. Okay, a couple more things. Okay, so going back to this idea that nerves and muscles evolved either separately or the same. So one of the predictions that comes about is if you think nerves and muscles evolved twice, there are a couple things here. It could be that nerves and muscles evolved independently in ctenophores and in Cnidaria and Bilateria. If that's the case, you might expect that there would be differences in the nerves and muscles, that there might be differences in the mechanisms or the ultrastructure that make them work. If nerves and muscles evolved once, either with ctenophores being over here and they evolved once, or they evolved deep in time and were lost in Porifera and Placozoa, you might expect there to be a lot of similarity in the mechanisms. Of course, there can be diversity and things change over time, but that's sort of an assumption you're going to make, is that if they evolve together, that there are common mechanisms there. In the genome papers that came out, people looked at this in great detail, and again, mainly focusing on nerves. So if we look at ctenophores, ctenophores lack a lot of the neurotransmitters that we see in cnidarians and bilaterians. They just don't have them. Remember, sponges and placozoa don't have nerves, nor do choanoflagellates, right? So a lot of the very conserved neurotransmitters we see in cnidaria and bilateria are absent in ctenophores. One thing we do see that's interesting is that ctenophores do seem to have a lot of, receptors for glucine. And that is actually involved in nerves and how nerves work and how nerves react. The interesting thing is we know that this can be a neurotransmitter and they have a lot of receptors for it. But importantly, they show a diversity in this type of neurosecretory proteins that we do not see in other animals. The bottom line here is it looks like their nerves are made a little bit differently. Okay, one more comment about that. So what does it take to be a nerve? Well, there are different types of nerves, right? There are nerves that are electrical, there are nerves that release neurotransmitters. But to be a nerve, you need to have a voltage drop across the membrane, and you need to be somewhat polarized and have some type of orientation. There's actually a ton of examples of other cells which are polarized and the cells have a polarity to them. We see that actually in a lot of protozoans. It's not to the same degree, it's not as strict, but the idea that nerve cells have properties that are totally unique to nerve cells in terms of having a voltage drop across the membrane is not true. They just have it to a greater extent than other organisms. But they're also using different types of genes. So this is just sort of a summary of, if we think ctenophores are sister, we have things like defining all of animals being the formation of collagen and gap junctions, which is a type of junction between cells. We don't see microRNAs or innate immunity in ctenophores, but we do see it in these other groups. And so, there is some logical progression here of how animals have developed and what they need to develop. Okay, so just to wrap up, this is sort of a larger question I've come to sort of deal with. Why are some phylogenetic results readily accepted and others considered incorrect, even when the methods are comparable, right? So there have been other major changes in animal phylogeny which people readily accepted and took up quickly. But this one is a particular hot button issue, and it's in part because people are very entrenched in their ideas. At least since I've been doing this type of stuff, there have been several major changes. Things like whether arthropods were with annelids or nematodes. This was a big argument, and a lot of people, because of segmentation, for years hung on to this, even though it was clearly incorrect. Brachiopods, bryozoans, and phoronids, did they belong on the deuterostome side, closer to the echinoderms, or were they closer to mollusks and annelids? people accepted pretty quickly, I think, that these guys were protostomes, but it's only because there had been other evidence before suggesting that. Also, people were pretty easy to accept the idea that platyhelminths did not include aceals, although there is a lot of argument, or there was a lot of argument over where the aceals fit in. Annelids have consumed a number of different phyla, and there's pentastomid, which I'll skip, but the last big one are cephalochordates, and whether cephalochordates were sister to chordates or whether sea squirts were sister to chordates. So some of these ideas people took up and were readily accepted. For whatever reason, the sponge debate has been a much, the sponge ctenophore debate has been a much more active debate and has caused a lot more, I guess, anxiety with people. I've been talking a little long. I'll just give a shout out to people who funded me, NSF, NIH, and collaborators. And I'll stop there and let's have some discussion.
Thank you very much, Ken, for the wonderful talk. I also would like to thank people that are on YouTube watching us. Thank you very much. I hope you enjoyed the talk as much as we did. Então, muito obrigada a todos que estão nos assistindo no YouTube. E a gente encerra por aqui a transmissão. Ok.